speaker, which is Daniel Müller-Komorowska, and he is a PhD student at the University of Bonn in Germany. And we will see, let's say, a kind of continuum from the previous talk, in the sense that we will look at some GABAergic interneurons and hippocampus. So Daniel, the floor is yours.
All right, thank you very much for this introduction. As said, my name is Daniel Müller-Komorowska, and today I'll be talking to you about our work on interneuron diversity in hippocampal CA3, and hopefully convince you that we have discovered a new interneuron subtype that co-localizes both somatostatin and VGLUT3. But first, about interneuron diversity more generally. Interneurons are, of course, a subtype of neuron that inhibits other principal cells. And these interneurons are very diverse, and they are so on three different categories. First of all, they can have very different electrophysiology. So they exhibit different firing behavior. They can be very regular firing, or they have very irregular firing, just as examples. They also can receive different inputs and send out different outputs. For example, here in green, we have a cell that receives both feedback drive, but also feed forward drive. In orange, on the other hand, is a cell that receives almost only feedback excitatory drive. So these cells participate in very different circuit motifs, and they also send their outputs to another set of principal cells. Third, interneurons are also diverse regarding their molecular markers, as we just heard a lot about. And there's a lot of methods to get at the molecular markers. One of the most common ones right now is transcriptomics, which oftentimes appears to you in the wild as a color map like this. And if you take these three criteria to talk about interneurons, there's, of course, hundreds of different interneuron types in the brain, but one of those that's somewhat interesting in the hippocampus is the cholecystokinin positive basket cell. And this one is electrophysiologically fast spiking, so fast firing, but it also adapting. It receives mainly feed-forward drive, and its molecular markers are cholecystokinin and VGLUT3, the vesicular glutamate transporter number 3. And recently, the McBain group has a paper where they show that VGLUT3 is not only a marker for these cells, but also these cells release both glutamate and GABA onto principal cells with both excitatory and inhibitory effects. So we were interested in uncovering potentially other cell types other than the cholecystokinin positive basket cell that also co-localize VGLUT3. And we found the somatostatin, a subset of somatostatin cells co-localizes VGLUT3. This work we've been doing together with Eric at the laboratory of Professor Zeinab Esmaeilpour. And here you see the green arrows mark two cells that have somatostatin antibody signal, somatostatin mRNA signal, and this is visualized with the RNA-Seq technique. And they also have VGLUT3 mRNA. And if we quantify this per layer, we see that this is predominantly, these cells are almost exclusive to CA3. So in CA3, there is about 30% of somatostatin positive cells co-localize VGLUT3 mRNA. So we wanted to characterize these cells, and the only way we saw to characterize them was to use a technique called Patch-Seq, which gathers both electrophysiological and transcriptomics data from the same single cell. I will first talk about the electrophysiology of that. So because we're interested in somatostatin and VGLUT3, we used mice that express Cre under the somatostatin or VGLUT3 promoter, and we use viral injections to get EYFP into those cells so we can visualize it. And then here's an example from an electrophysiology recording of an SST positive cell and a VGLUT3 positive cell. We can patch those cells in the three layers of the hippocampus, stratum radiatum or stratum oriens. But as a control, I also patched a lot of cells in the pyramidal cell layer, and I call those putative PCs, pyramidal cells. And from each of those recordings, we can quantify 28 different electrophysiological properties. So first of all, then, of course, it's interesting to do dimensionality reduction on those 28 electrophysiological parameters. And the first finding was that this was anticipated, but there is a clear cluster that contains all the cells from the pyramidal cell layer. So these are the putative pyramidal cells. So it was good that I patched those, because this tells me that there's also some that were in the stratum oriens and stratum radiatum that have pyramidal-like properties. So we throw those out because we want to get more into the interneurons, recluster them, and the electrophysiology alone does not form very well-defined clusters of these interneurons. So then we need to move on to the transcriptomics. So these cells here, these are 155 cells that I patched, and out of those we could get cytosol of sufficient quality, mRNA quality from 97 cells, and these are shown here. And first finding was, do we also find the somatostatin and VGLUT3 co-localization in the transcriptomics data? And the answer was yes. Out of these 97 cells, 12 are both somatostatin, and SLC17A8 is the gene name for VGLUT3. So these cells are both somatostatin and VGLUT3 positive. And then we looked at some known interneuron markers to see if any of those characterizes these co-localizing cells. And interestingly found that tyrosine hydroxylase, it was the one that significantly can classify these co-localizing cells. And by the way, these transcriptomics work we're doing with the precise core facility that exists here in Bonn, which have done great work with our samples. So tyrosine hydroxylase is a putative marker for these co-localizing cells. But now that we have the transcriptomics data, we can look again at the dimensionality reduction and annotate the co-localizing cells. And unfortunately, neither the transcriptomics nor the electrophysiology embedding the colocalizing cells form well-defined clusters. And we've not only done the dimensionality reduction, but we've also, of course, done more traditional statistical tests to see if any electrophysiological parameter defines these colocalizing cells, and we found none of our 28 parameters does so. So something that I'm working on right now, and that's more directed towards my scientific future, hopefully, is that here you see an example of one electrophysiological parameter, which is called maximum frequency. In the middle, you have the co-localizing cells. See no statistical difference between these groups, but even within the groups, there's a pretty wide range. So you can have around 100 hertz between cells within a group. So I'm interested in modeling this. So I've started to use the Brian 2 model fitting toolbox. And here you see these are recordings from one cell, and sweep 1 is a very low current injection that in blue you see the data, in orange the model. And this model that I fitted captures that there's only 3 spikes here, and they are all only in the beginning. And then at sweep 3 at much higher strength, we get a more sustained high firing frequency. And then I want to later use these kind of models to get a biologically plausible model of CA3 that incorporates the diversity of different interneurons. So in summary, a subset of hippocampal CA3 interneurons co-expresses somatostatin and VGLUT3. Patch-Seq allows for the characterization of such rare molecular subtypes of interneurons. Somatostatin and VGLUT3 co-localizing cells are not electrophysiologically unique. And tyrosine hydroxylase is a putative marker of these co-localizing cells in the hippocampus. With that, I want to thank you for your attention. Thank my supervisor, Heinz, Sandra and Eric for the great work on the RNA scope, Christian and Heidi from the core facility that have done great work with our RNA samples, my wife, who always helps me improve these presentations, and my presentation, my figures in general, and also the Neuromatch organizers and the community. I'm somewhat of a Neuromatch veteran right now. I was at the first one, the second one, IT8 at Neuromatch Academy, and it's pretty great to kind of grow with the community here. And if you want to follow up on the Interneuron grind, my Twitter and my email, and contact me.
Thank you very much, Daniel, for the wonderful talk, and also for your constant presence at Neuromatch. So one of the early adopters of the Neuromatch idea, and that's great to see. So if you have questions for Daniel, please write them there in the Q&A. And we have maybe one from Elena. Do you think it is possible that instead of there being separate clusters for which we have not found the parameters to define them, there is a continuum that might be more dynamic than we realize? For example, cells changing class, for example.
Yeah, so I'm personally, I subscribe to the continuum theory of interneuron diversity. There is some work in the dentate gyrus from the Bartos lab that claims that they show, they provide evidence against the continuum hypothesis. So they use electrophysiology, molecular markers, and morphology. And using all three of those, they are able to get clustering. But in the end, you can always get clustering, but the question is, how meaningful is this clustering? So it's hard to say. I'm not perfectly convinced that their clusters are the end all of interneuron diversity in the dentate gyrus. And I think even if there are clusters, they can still be they can exist. Clusters and continui can exist, I think. And I'm personally, because I had so many clustering failures, as you can see, I'm more on the side right now for CA3 that it's a continuum. Yeah, that's
okay.
I think that's a good question, and right.
And we have a last question. So from Jason, SST positive neuron in hippocampus tend to target dendrites. Do these SST VGLUT3 neurons show similar targeting?
I would love to know that. And we didn't really have a method yet to specifically get an opsin only into the colocalizing cells. We can get fluorophores into somatostatin cells. We can get them into VGL3 cells, but getting fluorophores only into these colocalizing cells, we are trying now a bidirectional genetics with SST flip recombinase and VGL3 Cre recombinase, and hopefully this will tell us, but we don't know yet. But this is definitely something that, as I said, input-output is a major criterion for interneuron diversity. So this is next on the table to kind of find that out.
Okay, and we have a very last question from Jan. Cool talk, I agree. What might be a functional role specific to those SST VGLUT3 co-expressing cells?
I don't know yet, and it's hard to speculate. We'll be doing some knockouts of the VGLUT3 in these SST cells, and do some behavior to see maybe if it does something to memory. But what specific? I mean, there are cells that are CA3 specific, so probably something sequence related or ensemble related or attractor related, but yeah, beyond that, I'm not really sure. Generally, though, if maybe they also co-release glutamate, that would be cool. But the functional role or the role of glutamate co-release is not actually known computationally. So if this would be known, though, that would be really cool. But I don't know yet.
Thank you very much, Daniel, for your answers.